This is Ritik, your biology subject matter expert from MEMONEET. And in this video, we are going to concentrate on the chemiosmotic hypothesis. So before we got to know like how exactly the photosynthesis takes place and what are the mechanisms that comes under photosynthesis. So today we are going to understand in depth regarding the mechanism and what all the process that happens during the photosynthesis, during the aspiration and all. So in chemiosmotic hypothesis, it actually explains about the mechanism of ATP synthesis in the chloroplast. So why ATP is very important? Because ATP is the one thing where it's a kind of energy, right? So because of the ATP, we do get energy. Like whatever the process that happens, the mechanism that happens, the cell, whatever they process, right? For everything, ATP is required. So chemiosmosis is nothing but a movement of the ions across the semi-permeable membrane. So here the semi-permeable membrane is actually thylakoid membrane. So it occurs in the chloroplast as well as in the mitochondria. See, when I'm talking about the chemiosmotic hypothesis, the chemiosmosis needs a membrane. So once it has a membrane, what happens with the membrane? It also requires a protein pump. Then the proton gradient. So the proton gradient is something through which the protons, they carry out, they move, and the ATP synthesis. The splitting of water takes place on the inside this membrane, that is inside this thylakoid membrane. So the protons accumulate in the lumen of the thylakoid. In the chemiosmotic hypothesis, splitting of the water molecules takes place on the inside of this membrane. That is what I was saying you, the thylakoid membrane. Then the protons or the hydrogen ions that are produced by the splitting of water, right? So within the lumen of the thylakoid membranes, that means all the protons or the hydrogen ions which got splitted by the water, so they will be accumulated the way you are observing here inside the thylakoids. As electrons move through the protosystem, okay, so what happens? The protons are transported across the membrane, and this happens because the primary acceptor of the electron, which is located towards the outside of the membrane, okay, they transfer its electron not to an electron carrier, but to the H carrier. Hence, this molecule removes a proton from the stoma while transporting an electron. This is very important. So when this molecule passes on its electron to the electron carrier on where inside this thylakoid membrane, the proton is released into the inner side of the lumen, okay, of the membrane. So as a result, one thing we will understand is the NADP reduce. See, what happens is here we have this thing called NADP reductase enzyme, which is located on the this region, that is the stoma region of the membrane. So what happens along with the electrons that comes from the acceptor of the electron of PS1, the protons are necessary for reducing of this NADP to NADPH plus H plus ions. These protons are also removed from the stoma. So whatever you're observing here, this procedure I am explaining. Okay. So from NADP to what happens here? NADPH plus H plus ions. So these protons has to be removed. Okay. Also has to be removed now. Hence, with the chloroplast, the proton in the stoma decrease in number, while in the lumen, that is here what happens, there will be the accumulation. You're going to see the protons and H plus ions accumulation will be more. This creates a proton gradient across the thylakoid membrane. So one thing will be clear that it is going to create a proton gradient across the thylakoid membrane as well as a measurable decrease in the pH of the lumen. So the gradient is very important. So why this gradient? Because it is very important. Why? Because it is the breakdown of this gradient that leads to the synthesis of the ATP. So if inside whatever the gradient, the proton gradient that we are seeing, right? If they got breakdown, then only we are going to absorb the synthesis of what? ATP. So the gradient is broken down due to the momentum of the protons across the, as I have explained you, because of the momentum of the proton, where across the membrane to where to the stoma, that is outside region, stoma through the transmembrane channel of CFO is actually a channel. So if that happens, then only you are going to absorb the release of the ATP synthesis. That is, ATP is going to synthesize. Then this ATP synthase enzyme consists of two parts here. One is CFO, one part actually will be the CFO and the other part will be CF1. See, in CFO is embedded in the thylakoid membrane. That is, it is actually this one. Okay. So it is present in the thylakoid membrane. Uh, yeah. So this one, okay, CFO is actually embedded, that is present in the thylakoid membrane and forms a transmembrane channel that carries or it actually facilitates the diffusion of the protein. So here you can see the proteins are diffusing across the membrane. Then we have another thing that is called CF1, this one. So here, yeah, this is the CF1. So here what happens on the other portion, which is called CF1, that what are the proteids that comes out of the surface of the thylakoid membranes or the protons which actually comes out of the surface of the thylakoid membrane? So that has to face on the other side faces the stoma, like they have to come to the stoma region, right? So this is stoma region. So once it comes to the stoma region, the breakdown of the gradient provides enough energy to cause a conformational changes of this in the CF1 particles of the ATP synthesis. Okay, which makes the enzyme synthesis several molecules of energy packets of the ATP. It's very complicated. Only thing that you have to understand is the representation. So that means in general, this entire schematic one part, this is what you have to remember. So if you remember the structure and where exactly what will come, then only you'll be able to understand the entire mechanism. If not, no. So one easiest method is instead of, mug upping the sentences and all, just try to recall and remember this diagram and try to know where exactly the labeling will come and its function. Then only you'll be able to understand the entire concept. Okay. So, in summarized, energy is used to pump the protons across a membrane to create a gradient or the higher concentration of the protons within the thylakoid membrane. This one we have discussed. Then the ATP synthesis has a channel that allows the diffusion of the protons back across the membrane. Okay. So as a result, what happens? This releases the enough energy to activate ATP synthesis enzyme that catalyzes the formation of the ATP. That means it forms the ATP along with the NADPH produced by the momentum of the electron. What happens like in this? You have seen the momentum of the electron. The ATP will be used immediately in the biosynthetic reaction taken place in the stoma, responsible for the fixing of the carbon dioxide and synthesis of the sugar. Moving to the next topic, it is all about what? The Kelvin cycle. So Kelvin and his co-workers, they worked out on the whole pathway, okay, and showed that the pathway operated in a cyclic manner. That is entire pathway, that is C3 pathway, Actually, it goes with the cyclic manner. So where the major thing is RUBP was regenerated. Okay. Ribulose biphosphate was actually what? It is regenerated. That is what he has explained by studying the entire cycle. Then the Kelvin cycle can be described under three stages. So one is carboxylation, then reduction, then regeneration. So let us understand the cycle one by one. Coming to the carboxylation, as you can see the figure here, yeah. Carboxylation is a fixation of the carbon dioxide into a stable organic intermediate. So what happens here is in the carboxylation, so this is the carboxylation. So carboxylation is a very important and a crucial step in the Kelvin cycle. So where this carbon dioxide, it is being utilized for the carboxylation of RUBP. Okay. So this reaction is catalyzed by the enzyme called as RUBP carboxylase, which result in the formation of two molecules of 3-PGA is nothing but 3-phosphoglycerate. So here what happens is this carboxylation in presence of RUBP enzyme. So you're going to observe the formation of 3-PGA. Okay. Since these enzymes also have a oxygenation activity, it would be more correct to call it as RUBP carboxylase oxygenase or RUBPISCO. Okay, there's nothing but RUBP carboxylase oxygenase. After the carboxylation, you will see the reduction. So in reduction, what happens? There are series of reaction that leads to the formation of one thing that is called as glucose. So the steps which involves here is utilization of the two molecules of the ATP for phosphorylation and two of NADPH. Okay, so whatever ATP and NADP. So this they are going to utilize in this reaction for reduction per CO2 molecules fixation. Okay, as a result, what happened? The fixation of the six molecules of the CO2 and six turns of the cycle are required for the formation of one molecule of the glucose from the pathway. As a result, you will see triose phosphate. Okay. And as a result, triose phosphate is one which is going to release the sucrose and starch, glucose. Then we have after this, you will see the regeneration. So in regeneration, what happens? The CO2, okay, of the regeneration of the carbon dioxide acceptor molecule, RUBP, is crucial if the cycle is to continue without any interruption. Then the regeneration steps require what? So it requires an ATP. So this is the end product. So if you need a regeneration step, again you require an energy. So for the energy you require, that means ATP is required for the phosphorylation. Again, once again the phosphorylation if you want to see. Then for that, it is required to form the RUBP. That is this one. Okay. So as a result, that's what I'm saying. So the end product will be the formation of the RUBP. That is ribulose 1,5 biphosphate. So this is about the C3 cycle or the Kelvin cycle. Hence, for every CO2 molecule entering the Kelvin cycle, three molecules of ATP and two molecules of NADPH are actually required. So it is probably to meet this differences in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. Clear to you? So to make one molecule of the glucose 6 turns of the cycle are required. So what comes out of the Kelvin cycle? So what comes in and what comes out? For example, if six carbon dioxide is being which comes in a Kelvin cycle, so will releases what? One glucose, this thing. Then 18 ATP will reach to 18 ADP. Okay, this one. And 12 NADPH will lead to 12 NADP. Okay. So this is all about what? C3 cycle. So in the coming video, we will be concentrating on the next other part, that is the C4 cycle and the factors which are going to affect the photosynthesis. Till that, if you feel any doubt, you can reach me out to the comment section and don't forget to like, subscribe the channel, and also please share with your friends. Thank you.